Imagine taking a state-of-the-art drug, right? A drug specifically designed to target and kill your cancer, only to find out it's simultaneously disarming the very immune cells trying to save your life.
Yeah, you're essentially paying for friendly fire, on a molecular level.
Exactly. It's wild. So today we are diving into the absolute paradox of pancreatic ductal adenocarcinoma, or PDA. We've got this massive stack of data from a 2026 study.
Right, the Tang L study from Cancer Center.
Yeah, that's the one. And it fundamentally challenges how we think about targeted therapies. The mission for today's deep dive is to explore this newly constructed cell type resolved prognostic atlas for pancreatic cancer.
It is just a remarkable piece of research.
Yeah.
I mean, the data set alone is staggering. We are looking at a cohort of 152 human patients profiling over 1.2 million individual cells using single nucleus RNA sequencing.
Wow.
And then they physically mapped another 3.1 million cells using spatial transcriptomics. What this team has done is basically move us past the era of treating a tumor as just, a monolith.
Right, they built an interactive platform too, right?
Panda.
Yeah.
CT Panda lets you navigate this new map.
Yeah.
And in the process, they've uncovered exactly why pancreatic cancer has such a dismal 13% 5-year survival rate.
Right.
It's notoriously treatment refractory, and this data finally shows us the physical and chemical infrastructure driving that resistance.
Okay, let's unpack this. Because to really grasp the sheer scale of what they accomplished, we have to talk about what standard oncology research has been doing wrong, or well, at least doing inefficiently for the last couple of decades.
Yeah, we've heavily relied on bulk-level transcriptomic sequencing.
Right. And I always think of bulk sequencing like trying to figure out what's wrong with the blended fruit smoothie.
A smoothie.
Yeah, like if the smoothie tastes way too sour, you have absolutely no idea if it was the strawberries, the kiwis, or the apples that ruined it. You just taste the net average. You need to taste each fruit individually, right?
That is actually a highly accurate way to look at it. In bulk sequencing, you take a tumor biopsy, which is a complex ecosystem of cancer cells, immune cells, blood vessels. You grind it all up, extract the RNA, and sequence it.
So you just get the average.
Exactly. You might identify a specific gene that's highly expressed in patients who die quickly, but because the sample is homogenized, you don't actually know which cell in that tumor is expressing the gene.
Is it the cancer cell mutating? Is it a macrophage malfunctioning? You just don't know.
Right. And to solve this, the researchers use single-nucleus RNA sequencing, or snRNA-seq, on the FUS-CC SNPDA cohort.
Now, wait, slow down for a second. Why single nucleus specifically? Why not sequence the whole intact cell?
Well, it mostly comes down to logistics and tissue viability. To do whole cell sequencing, you generally need fresh tissue. The cells have to be alive.
Oh, I see.
But gathering a large enough cohort of fresh pancreatic tumors and then waiting 5 to 10 years to see which patients survive, I mean, that takes decades. By targeting just the nucleus, the researchers could use frozen archival tissues.
Oh, wow.
Yeah, they went into the biobank at Fudan University Shanghai Cancer Center and pulled samples from 152 treatment-naive patients whose clinical outcomes were already known.
That makes total sense. It's like a biocomputational time machine. Because PDA progresses so aggressively, what was it? Over 80% of the patients in this specific cohort had already reached the clinical endpoint?
Exactly, 80.3%. They had passed away.
Right. So we already have the robust long-term overall survival data. We know exactly who lived and who died, and now we can look at the tax returns of their individual cells from years ago.
And when they looked at those individual readouts, they categorized 23 main cell types. For the first time, they bridged intratumoral gene expression directly with patient survival.
Which is huge. So just to give a quick recap for you listening, we've moved from grinding up whole tumors like a smoothie to analyzing 1.2 million individual cells, linking specific cell types directly to patient survival. But have you ever wondered why a cancer drug might work perfectly in a lab petri dish but completely fail in a human body? Well, the answer lies in what these specific cells are actually doing. Which brings us to the next massive finding.
Yes, the CRPRGs, cell type resolved prognosis-related genes. They identified over 7,000 of them.
And this is where the data gets deeply, deeply unsettling. Out of those thousands of genes, they flagged 619 of them as BiPRGs, right? Bidirectional prognosis-related genes.
Correct.
I need you to explain this mechanism because, I mean, the idea that a single gene can be playing both sides of the war is mind-bending.
It really is. It upends all our traditional pharmacology models because a bi-PRG is a gene that has completely opposite effects on a patient's survival depending entirely on which cell type it is operating inside.
So the gene itself isn't intrinsically good or bad?
Exactly. Its function is entirely context-dependent. They are the double agents of the tumor.
Okay, give me a concrete example. How does a gene know which team it's playing for?
Let's take the ITGB1 gene. It encodes integrin beta-1. Now, integrins are transmembrane receptors. They help cells stick to the surrounding tissue, the extracellular matrix.
Okay, so they're like cellular anchors.
Right. If ITGB1 is highly expressed in the actual PDAC cancer cells, it is highly detrimental to the patient. It allows the cancer cells to anchor to the stroma, driving tumor proliferation, invasion, and ultimately drug resistance.
So if I'm a pharmaceutical developer looking at that data, I'm thinking, great, I found my target. I'm going to make an ITGB1 inhibitor, flood the tumor, block the cancer cells from anchoring, and stop the invasion.
Which is exactly what you would do if you were only looking at bulk sequencing. But the single-cell data reveals the trap. That exact same ITGB1 gene is heavily expressed in the patient's T cells.
Oh, no.
Yeah. And in a T cell, integrin beta-1 is essential for motility. The T cell uses it to physically navigate through the tissue to reach the tumor and maintain its anti-tumor function.
Cool. Wait. So a drug targeting a bad cancer gene could accidentally be disarming the patient's immune system at the exact same time.
Precisely. If you use your blanket ITGB1 inhibitor, you detach the cancer cells, sure, but you simultaneously paralyze the patient's killer T cells so they can't even enter the tumor bed.
That is terrifying.
And we actually see this happening in real-world clinical development. Look at the ADM17 gene. High expression in tumor cells means a poor prognosis.
So it's bad for the patient.
Yes, but in dendritic cells, those are the antigen-presenting cells that prime your immune system to recognize the tumor. ADM17 is highly protective.
Let me guess, we're already trying to inhibit it.
Yes. Currently, there are clinical trials exploring ADM17 inhibitors like adagrasib. This study provides a massive warning sign. While adagrasib might inhibit the tumor, it could accidentally shatter the dendritic cell's ability to initiate an immune response.
Wow. Okay, so to recap this part for you guys listening, genes are not inherently good or bad. Their role depends entirely on the cell they inhabit, which makes blanket targeted therapies really dangerous. So if we can't just carpet bomb a gene, how do we understand the way these cells manipulate each other? Because clearly they aren't operating in isolation.
They're highly coordinated. And to understand that, the researchers divided the patients into two extremes: short-term survivors living less than a year, and long-term survivors living over 5 years.
STS and LTS.
Right. Then they mapped the ligand receptor interactions, basically the chemical communication pathways between all the different cell types in the tumor microenvironment.
The cellular chat network, who is talking and who is listening.
In the short-term survivors, the network was highly active. But here's the twist. The cancer cells themselves weren't the primary broadcasters.
Really?
No, the malignant cells were acting as receivers. The dominant senders were the non-malignant structural cells, specifically the pericytes, endothelial cells, and the cancer-associated fibroblasts, or CFs.
CFs. I was absolutely fascinated by the role of CFs in this study. If the tumor is like a corrupt city, the CFs are the corrupt city planners.
That's a great analogy.
They aren't the ones pulling the trigger, but they are laying down the fibrous highways, all this extracellular matrix deposition, specifically so the cancer cells have the physical infrastructure to easily invade the bloodstream.
And it goes far beyond just physical infrastructure. The CCAPs are actively dictating the phenotype of the cancer. The data shows keycaps heavily secreting a protein called activin A.
Which is encoded by the INHBA gene, correct?
Correct. When the PDA cells receive that activin A signal, it triggers a catastrophic phenotypic shift. The tumor cells transition from a classical state into a highly invasive basal-like state.
So they're essentially downloading a lethality upgrade from the local environment.
Basically, yes.
But it isn't just chemical signaling. The actual physical construction project the CCAPs are running, this intense desmoplasia and fibrosis, it stars the neighborhood of oxygen, right?
Yes, the dense fibrous tissue compresses the local blood vessels, creating a state of profound hypoxia. And this is where we see the metabolism of the tumor fundamentally alter.
To survive the famine?
Exactly. The lack of oxygen activates HIF-1 signaling pathways.
Hypoxia-inducible factor 1.
Right. HIF-1 acts as a master transcriptional regulator. It pushes the cells away from normal oxidative phosphorylation and heavily into glycolysis. They aggressively start converting pyruvate into lactate to generate energy without oxygen.
That's so insidious.
And the researchers found this specific metabolic shift is universally linked to lethal outcomes across multiple cell types in the short-term survivor cohort.
Okay, quick bullet recap. Short-term survival is driven by intense chatter and physical infrastructure built by non-cancerous structural cells, the CFs, that help the tumor escape.
Perfectly summarized.
So if you're listening and you picture the tumor as this corrupt city, what does the absolute worst neighborhood in that city actually look like physically?
Well, to answer that, we have to look at the spatial transcriptomics data. Single nucleus sequencing gives you incredible detail, but you have to dissociate the tissue to get it. You lose the geography.
They scramble the map.
Exactly. So to map the actual neighborhood, they used a high-plex technology called Xenium in situ 5K.
Walk me through the actual physical reality of how Xenium works on a slide. Because you aren't grinding anything up here.
No, you're taking a microscopic slice of the tumor tissue, right, and leaving it completely intact on a glass slide. Instead of extracting the RNA, you apply thousands of fluorescently labeled probes directly to the tissue.
Oh, wow.
These probes hybridize or bind to specific RNA transcripts right where they sit inside the cells. You then use high-resolution imaging to capture the fluorescent signals. They did this for 3.1 million cells across the physical space of the tumors.
So you can calculate exactly which cells are standing shoulder to shoulder.
Precisely. They identified distinct cellular neighborhoods, or CNs, and they discovered one specific physical cluster that is highly lethal, CN6.
Here's where it gets really interesting. What makes CN6 so lethal?
It's defined by a very specific triad of cells existing in close physical proximity. It consists of short-term survival PDA cells, cancer-associated fibroblasts, and monocytes or macrophages.
The PMC triad.
Yes.
The absolute worst neighborhood in the city. You've got the mob boss, the corrupt city planner, and a hijacked immune cell, all sharing the exact same block.
It's a devastating combination.
Yeah.
And we know definitively that this physical neighborhood drives chemotherapy resistance.
How do we know that for sure?
Because they correlated these spatial maps with clinical grading. Specifically, the RECIST criteria, which evaluates how a solid tumor responds to therapy.
Okay, so measuring if it shrinks or grows.
Right. In patients who achieved a partial response, meaning the chemoimmunotherapy was actually working, the researchers saw the physical disruption of the CN6 neighborhood. The triad physically shrank and broke apart.
But if the treatment was failing...
In patients with stable or progressive disease, CN6 was deeply entrenched.
They also observed some incredibly sinister physical structures when the disease was progressing. I was reading that section, and they found that the tumor cells were physically wrapping themselves around intratumoral nerves.
Nerve microinvasion, yes.
Yeah.
It's one of the most agonizing hallmarks of pancreatic cancer.
Literally crawling along the nerves.
Exactly. The tumor cells use the neural tracts as alternative highways to escape the primary tumor site and evade the hostile hypoxic environment.
Right.
It's a major driver of tumor recurrence and intense neuropathic pain for the patient.
That is horrifying. So let's recap again. Using spatial mapping, researchers identified specific physical clusters of cells, like the lethal CN6 neighborhood that drive chemotherapy resistance.
Yes.
Which puts you in the shoes of an oncologist looking at this data. With millions of data points and countless neighborhoods, how does a doctor actually use this to cure a patient? If almost every gene plays a dual role, how do you find a target that won't blow up in your face?
And that was the ultimate goal of building the HadoPanda platform. It's a free web platform for anyone to explore this data. They used it to search for a holy grail target.
A gene that is universally bad across multiple cell types.
Exactly. Because if a gene drives poor prognosis in the cancer cells, the fibroblasts, and the macrophages all at the same time, inhibiting it circumvents the double agent problem we discussed earlier.
And their algorithms spat out a gene called PLOD2. And honestly, when I dug into the biochemistry of what PLOD2 does, it reads like a masterclass in biological defense systems.
It really does. PLOD2 encodes an enzyme responsible for creating cross-links between collagen fibers.
Basically weaving them together into this incredibly dense, rigid matrix.
Right. Pancreatic tumors are notoriously hard to the touch. They feel like a rock during surgery. PLOD2 is a primary driver of that stromal stiffness.
But the genius if you can even call it that, of the tumor, is that the stiffness isn't just passive armor. The physical rigidity actually alters the internal machinery of the cells. The study details how this stiffness induces something called F-actin alignment.
Yes, within the cell's cytoskeleton, and that F-actin alignment triggers a devastating feedback loop.
How so?
Well, every cell has an internal garbage disposal system to regulate protein levels. For PLOD2, that disposal system relies on a ubiquitin ligase called TIM21. Normally, TIM21 tags excess PLOD2 proteins and tells the cell to degrade them.
But the stiffness messes that up.
Exactly. The stiffness-induced F-actin alignment physically impedes TRM21. It shuts down the garbage chute. So the PLOD2 makes the tumor stiff, and the stiffness protects the PLOD from being degraded. It's a vicious cycle.
A self-amplifying loop of impenetrable defense. So how do you break it?
Standard inhibitors aren't enough. The researchers engineered a highly sophisticated drug called PROTAC G3. PROTAC stands for proteolysis targeting chimera.
I love the mechanics of PROTACs. They act like molecular hitmen. It's not just a chemical wedge that blocks a receptor. Picture a microscopic assassin with two hands and a flexible chemical linker in the middle.
That's a great visual. One hand grabs the target protein, PLOD2. The other hand grabs that deactivated garbage disposal enzyme, TRIM21.
And it forcibly drags the PLOD2 into the garbage chute. It bypasses the F-actin blockade and forces the cell to digest the bad protein. You are completely wiping it out of the cellular inventory.
And the clinical evidence is striking. To ensure they weren't accidentally triggering an immune double agent scenario, they tested PROTAC G3 and CD34 plus humanized mice.
Wait, humanized mice, what does that mean?
These are immunodeficient mice engrafted with human stem cells. So they develop a functional human immune system, complete with human T cells and macrophages.
Oh, wow. So that's the ultimate proving ground. If the drug hurts human immune cells, it will definitely show up there.
Precisely. And the results. PROTAC G3 successfully degraded PLOD2 and dramatically reduced the stromal stiffness.
They dismantled the barricade.
Exactly.
Yeah.
And by dismantling that physical barricade, CD8 plus cytotoxic T cells were finally able to flood into the tumor microenvironment and do their job. The tumor shrank significantly.
And the safety profile.
Crucially, it did not cause weight loss or systemic toxicity in the mice.
That is incredible. Okay, let's bring it all together. Quick recap. By finding a universally bad gene, PLOD2, and using a targeted degrader, PROTAC G3, researchers successfully dismantled the tumor's physical defenses in living models.
That's exactly right.
So what does this all mean for you, the learner, and the future of medicine? We moved from looking at the blended average of a tumor to reading the individual state of millions of cells. We mapped the corrupt physical neighborhoods, and we used all that data to find a universal vulnerability.
It represents a fundamental shift in how we approach oncology. For decades, the strategy has been just a war of attrition. Apply systemic toxins and hope the tumor dies before the patient does.
Right, poison everything.
But this single cell and spatial data reveals that the tumor is just a highly adaptable ecosystem. If our own immune and structural cells have good and bad states depending purely on their environment and who they are communicating with,
then the future of treatment is reprogramming, not just poisoning.
Exactly. It's urban renewal. Reprogramming the bad neighborhoods and relocating cells to turn a lethal cancer into a manageable or even curable condition.
It completely shifts our relationship with the disease. And it leaves me with this final provocative thought for everyone listening. If physical proximity dictates how aggressive a cancer will become, could we theoretically take spatial biopsies of benign tissue?
Oh, absolutely.
Could we map the architecture of an organ and predict a malignancy based purely on the way fibroblasts and macrophages are clustering long before the first true cancer cell even mutates?
Predicting the fire by watching where the arsonists are gathering. That is exactly the frontier this data opens up.
And the most incredible part is that the raw data driving this shift isn't locked away in some corporate vault. The TIP Commandi platform is entirely open access. You can go online right now and explore the exact same cellular neighborhoods we just discussed.
It's an incredible resource.
It really is a profoundly different way of looking at a disease that has defied us for so long. Thank you for diving into the architecture of it all with us today. Check out the platform, ponder the idea of cellular urban renewal, and we'll see you on the next Deep Dive.